Thanks everyone for being here. This is our webinar. We're going to cover a lot of interesting topics today, so let me just get straight into the agenda. So we're really fortunate to have some of our world-renowned collaborators on the call today, and we're also very fortunate to have them as partners. I'd like to introduce them briefly, and then I'll pass it to them to give their talks. So starting with Dr. Margot Cousin, she's a medical geneticist and an assistant professor at the Mayo Clinic, where she leads the N of 1 therapeutics program at the Center for Individualized Medicine. She focuses on advancing gene-targeted therapies from lab to clinical trials, and her work involves various therapeutic areas and partnering with advocacy groups like ours, industry, and academia to bring solutions to the rare disease community. She's going to be talking to us today about the science and knowledge gaps around ADLD. After her talk, we'll have Dr. Anjana Sevagamoorthy. She's a physician and clinical research scientist at CHOP. She specializes in FDA-compliant natural history studies for rare leukodystrophies and supports academic industry partnerships to accelerate therapeutic development. At CHOP, she specifically represents the GLIA CTN and is involved in setting up the data-related infrastructure for the ADLD natural history study. She's going to be talking to us about real-world evidence-based longitudinal natural history studies in leukodystrophies. And then finally, we'll wrap up with Dr. Radhika Dhamija. She's a child neurologist and medical geneticist at the Mayo Clinic, where she serves as an associate professor of medical genetics and assistant professor of neurology. She leads the clinical care and research for patients with autosomal dominant leukodystrophy and plays a key leadership role in the ADLD natural history study, where she's focused on advancing diagnostics and longitudinal care for rare white matter diseases. I just want to share a couple quick updates from the ADLD Center, and then we'll pass it to Dr. Margot Cousin to get started. So for anyone who's joining us today for the first time, there's a little bit of context. About 5 years ago, after seeing generations of our family members affected by ADLD, a few of us relatives got together to ask, what can we do to bring an end to the disease? And that conversation led us on a journey of discovering a variety of other patient advocacy groups, communities, and many more ADLD patients, ADLD researchers, and physicians, including many of you who are here today and beyond. Building on what we learned through that journey, we started this nonprofit organization, the ADLD Center, and we're now engaging with patients in the USA, India, Brazil, Uruguay, and many other countries. We're also partnering with as many ADLD researchers as possible, and we're fortunate to have a few of them on the call with us today, as I mentioned. And as a result, we now know much more about the disease, how to manage it, and what kinds of efforts can help us get towards a treatment or a cure for it. So real quick, some exciting news. The first ADLD patient received their dose of a new therapy in March, and we can talk more about that during the Q&A if you have any other questions. We had also sent an email about it earlier, so there's more information in the email. If you haven't received it, just send a note in the chat, and we can add you to the mailing list. We kicked off the natural history study, and at least 15 patients have already consented to participate, maybe more. We'll have Dr. Radhika Dhamija and others speaking about that today. And we awarded a new grant to Dr. Jaspreet Singh of the Henry Ford Health System to investigate microRNA-based biomarkers for ADLD in plasma. So basically, we want to know if there's a treatment for ADLD, is there a quantifiable way we can know if it's working or not by taking a blood sample. So we're having Dr. Singh investigate different approaches to do that, one of which is microRNA based. So with that, I'll be passing it to Dr. Margot Cousin. I'll stop sharing my screen to kick us off.
Thank you, Bishan. I'm really excited to be here today. I'm just going to pull up our slides. Everyone see those?
Yep.
Perfect. All right, so as Bishan said, thank you for the kind introduction. I'm the director of our N of 1 therapeutics program in the Center for Individualized Medicine at Mayo Clinic. I'm a clinical and translational scientist and really trying to bridge the gap between basic science and clinical interventions, trying to improve care at the bedside with novel discovery. So I'm going to walk through some things you probably already know, but I want to set the stage for where we need to go. So we're going to start at the beginning. So what do we know about ADLD? And I'm not going to read all of these. but this is probably information you're already aware of, right? This is an adult onset disorder, often between the ages of 30 and 60 for onset, and was previously diagnosed in the first reports as primary progressive multiple sclerosis. Importantly, this diagnosis is can often be made clinically just through clinical features and MRI features, which is great since sometimes genetic testing is difficult to achieve. Slowly progressive and fatal, and the slowly progressive part makes it challenging to measure in clinical trials. So we're going to talk more about that. Ultra rare disease also has many challenges, as you all know, and likely underdiagnosed. So symptoms often begin with autonomic dysfunctions. This might include bladder or bowel erectile dysfunction, dizziness upon standing, heat intolerance, often followed by motor symptoms, spasticity, weakness, gait abnormalities, and cognitive symptoms may occur late in the disease. Brain imaging has a distinct pattern affecting the white matter, which is associated with loss of myelin, which we know is that protective covering of our neurons that helps keep them healthy and functioning. And importantly, ADLD is caused by duplications of the lamin B1 gene, or in rare cases, deletions upstream of that gene. So this is kind of where we're starting. I'm going to dive into the genetics because that's what I tend to do. So if we, all of us have, 23 pairs of chromosomes in our cells, and here's a schematic of chromosome 5 where the lamin B1 gene is. So if I zoom in there, you can see here's a very simplistic structure of the area. You can see the LMNB1 gene and a neighboring gene upstream of that in a normal individual. So in ADLD, the most common genetic mechanism of disease is duplication. So here you can see an example of sort of this tandem duplication of the gene. But we also have seen patients that have a deletion upstream. So now you can see the distance between this upstream gene, as I have listed here, and lamin B1 is shortened. So there's a regulatory region in here that also controls how this LMNB1 gene is expressed. And importantly, either case, whether it's the deletion or the duplication, leads to overexpression. So we get too much gene product being produced when we have either duplication or upstream deletion. So what does that do? So we know that the lamin B1 gene makes the lamin B1 protein, and this is associated with the nuclear lamina. So the nucleus of all of our cells has this membrane around the outside that keeps our DNA inside and protected, and lamin B1 is associated with that membrane region. And you can see in blue here, these little squiggly guys are the lamin B1 proteins, and you can imagine if we have way too many of these, you see all these other proteins around this nuclear membrane, and they do many, many functions, right? If we have way too much of this protein present, it's really making it difficult for those other proteins to do their jobs. So we think this is leading to dysfunction in many different ways, kind of downstream of the nucleus. So simplistic idea, but this is essentially where we're at, right? Too much of this protein is causing issues. So what are our goals, right? I mean, ideally, we want to have a specific and curative therapy, but we also, very simplistically, right, we want to improve disease management, we want to improve quality of life, and we want to better understand the disease course and be able to predict prognosis. So some of the things we've done to date to kind of allow you to understand kind of where we're coming from and what we've been working on. We have formed a collaboration with the N. Foundation for Therapeutic Development. We have started a ADLD Clinical Care Center at Mayo Clinic that Dr. Dhamija, who will be speaking later, is leading. We have established recommendations for clinical care and through literature review and expert panel consensus, and that manuscript should be forthcoming soon. We've hosted the second International Symposium on ADLD in Mayo Clinic last year. And what we're going to talk about today is launching this real-world data natural history study. And this is in collaboration with the Children's Hospital of Philadelphia and the Global Leukodystrophy Initiative with support from the ADLD Center and partnership. And that's what we're going to focus on. So to that end, what are the questions, right? Do we have a therapeutic target? And how do we know if an intervention is providing benefit? And these are the two things we need to move a therapy forward. So some beautiful work done by Elisa Giorgio and her colleagues really set the foundation of understanding of, can we correct this overexpression and would it provide benefit in diseased cells, right? In cells that are already affected. So they did, I'm not going to go into the details here. There's a lot of information on this slide, but essentially they were using an RNAi approach to knock down that overexpression. So really hitting that genetic mechanism of disease. And we're able to show that we could recover much more normal function in cells from patients that had ADLD. So this gave us confidence that actually targeting, the gene that causes the disease could be beneficial for patients. So, this has really led to us focusing on LMNB1 expression as the target for therapeutic development. And that work is ongoing. But what about the other question, right? How do we know if an intervention is providing benefit? Are there things we can measure, right? Blood tests, imaging, clinical evaluation, scales or surveys that we can measure to show benefit in a shorter amount of time than maybe just looking at MRI, for instance, that is really slow to change. And what do we need to control for? Do we subset patients based on other characteristics, such as age of onset or the type of genetic alteration they have? And when is the best time to treat in order to see maximal improvement, to think about how you would design your clinical trial and who you might enroll. So just simplistically, right, if we have a potential health parameter, right, so we're measuring overall health or function, we see that maybe there's this rate of decline in a patient over time associated with disease. And then without treatment, that might continue on looking as a progression rate like this. And treatment, we hope they would do better, right? We'd have this measurable difference of whatever this parameter might be. You can imagine if that rate of decline continued in the same manner in treatment as it did pre-treatment, we might think that that's not doing anything. But what if the actual rate of decline were to increase in that patient untreated, right? We would still have this measurable difference. But we would only know that if we really understood how that parameter changed over time in a really granular and specific way. So this is what we need to be able to do in a natural history study to be able to show that when we treat, we can really confidently measure benefit. I mean, the worst case scenario would be we have a treatment and we put it into clinical trial and we fail to measure a change, but we really believe that it's there, right? So this is important work to understand at a granular level, how do we measure change when indeed it could be present? So a little bit more about what we know. So Pinson and colleagues published this really beautiful study in 2015 and is the largest data set we have on longitudinal data for ADLD. They described 23 patients and followed them for up to 17 years, which is amazing. But it was 2 families and they all had LMNB1 duplication. So they, really tried to describe the disease in the many different aspects. And here you can see on the right, we're looking at this EDSS score is just a measure of severity of disability. So higher is worse. And you can see the age of the individuals over time, and there's a direct correlation, as these patients aged, they did had more significant disability. Similar was true for the changes in MRI. But again, this is over decades, right? So we can see a steady progression of disease. But is this really what we want to measure in a trial? It's going to be difficult to run a trial for decades. So other observations. So that study was of patients with duplications and just of 2 families. So not all duplications cause ADLD. We believe that the level of overexpression may be associated with the rate of disease progression. So perhaps the different types of genetic alterations might lead to changes in overexpression or variability in overexpression that might be related to outcomes, but we haven't studied that yet. And patients with the deletions upstream of LMNB1 also seem to have a little bit different clinical features. So it's important to understand what's the variability between these individuals. And I'm not going to read all of this, but this is sort of a summary of all of those questions. And these are all the types of questions that we really need to have concrete answers in order to, again, translate these therapies into trials, which can then get us into, clinically approved therapeutic solutions. So these are the questions that we want to answer with this study and with the future studies that will come from this initial one that's going to really inform us on how to move forward. I know we have a lot of slides today because we're excited to share a lot of content. So I'm going to leave it with this last slide here and then hand it over to Dr. Sevagamoorthy. So we've already launched this ADLD natural history study. This is a chart review study. So we're using real world clinical data that's already been collected for individuals and participants who want to, individuals who want to participate in our study can share that medical record data with us. So this allows us to maximize participation. Anyone can participate from anywhere. and allows us to collect a lot of longitudinal data in a short amount of time, right? We're collecting, years of data in the past as quickly as we can to generate that new knowledge. This will inform us on how to do a proper prospective natural history study that we would conduct in a similar way you would do a clinical trial that would then inform our actual clinical trial and help serve as control group. So that was a lot, but I'm going to hand it over to Dr. Sevagamoorthy, who's going to talk about the amazing resource they've built at the Global Leukodystrophy Initiative and the Children's Hospital of Philadelphia.
Thank you, Margot. Just one quick second,
Sure.
just because people have been joining, I just wanted to recap where we are, and then Anjana, sorry to jump in here. So we had a quick introduction, and then Dr. Margot, cousin from Mayo Clinic, has started us off. We now have about 10 minutes from Dr. Sevagamoorthy, and then another 10 minutes from Dr. Dhamija to talk about some of these areas, and then everyone will get an opportunity at the end around 11:40 to 11:45 to start Q&A, so that you can have some of your questions answered. If you do have questions, you can send them to me directly in the chat. You can put them to everyone in the chat, or you'll be able to have the opportunity to raise your hand. Sorry to take up your time, Dr. Sevagamoorthy, and I'll turn it back over to you.
Thank you, Inderpal. So nice to meet you all in this forum. Just a brief introduction. I'm Anjana Sevagamoorthy. I'm a scientist at the Children's Hospital of Philadelphia, and I also lead the GLIA-CTN Real World Evidence Core. And today next slide. Today I'm going to be, so I don't have any disclosures. Today I'm just going to quickly jump into like what the GLIA-CTN consortium is, and then jump directly into really the main topic for the discussion, which is what is a natural history study, what is our approach, and why is this important? Next slide. So GLIA-CTN is a consortium of institutions, clinicians, scientists, and advocacy partners with the goal who are all working towards promoting advances in the diagnosis and treatment of leukodystrophies, specifically to create a robust infrastructure to allow for collection, sharing, and analysis of longitudinal natural history data. Next slide. We currently have 11 active sites who are collaborating and enrolling patients into the study. Next slide. And we are working towards growing this. So we are adding another 11 sites and over 3,500 subjects have been enrolled thus far. Next slide. So jumping into what is a natural history study. Next slide. So natural history studies are pre-planned observational studies performed to track the course of the disease across the lifespan. There are two types of natural history studies. One is the concurrent. This is where when patients go to a healthcare provider, they perform assessments during the clinic visit, and based on that, they provide a score or, write a certain, assignment as well. So this is a gold standard since it's done real time at the point of visit. But it takes time, it is resource intensive, and by default it's patients who are able to present at the clinic on the specific date who are being assessed. So more severe patients who have logistical barriers or also practical barriers related to the disease are unable to participate in this study. And most importantly, assessments are performed after a diagnosis. So it's all post-diagnosis data that we're able to accumulate from this, and we learn less about the pre-diagnostic clinical information. This is where a non-concurrent natural history study comes into play. The data source is already existing, and so we're able to recruit a large amount of data in a very short time, and it's less impacted by rarity, and it's less burdensome to the families as well. And so using this, we're able to collect both pre-diagnostic, but also post-diagnostic data. So use of concurrent and non-concurrent natural history study is very important, especially when we're studying rare diseases like the leukodystrophies. Next slide. When we say data and real world data, we're referring to here are like medical records, claims data, product registries, disease registries, and digital health technologies like the wearables. And these are all not really data that's generated for research. So there are challenges associated with it when we try to repurpose it for research. some of which are highlighted here, and they are like heterogeneity in the quality and duration of medical records. There is unclear documentation specifically on the onset of medical events, and there is also data missingness. Specifically, when we think about using claims data, there is insufficient depth of the data, particularly, a code is assigned on a certain date, but that doesn't essentially refer to the onset of the disease or the symptom itself. Also, we don't have ICD codes for a lot of our rare diseases. This is a work in progress right now, but there's still, there's still a lot of work to be done on this. So we need a standardized approach to performing real world-based natural history studies. Next slide. And this is where we jump about like how we are performing that. Next slide. So we have developed standard operating procedures, just like how we will have in a clinical trial. So really mimicking a clinical trial epoch to ensure there's consistency, interoperability, and data quality and integrity throughout the process. And these standard operating procedures cover a lot of different topics, and they're version controlled as well. So it talks about all the way from like when a person is trained, like a research personnel training, to patient enrollment, how the data data sources should be collected, how natural history eligibility is determined, how we calculate the ages, dates, and also how we perform data entry into REDCap or Part 11 databases, how the data is going to be managed, and how it's going to be monitored too. Next slide. This is an overview of the GLIA-CTN approach to determining natural history eligibility. So patients are recruited from multiple sites. Once they're enrolled, consented and enrolled, we create study IDs, and records are collected and stored in the REDCap database. Then our TCs go in to review the clinical documents, including MRIs and genetic reports and biochemical reports as well, if available, to determine if the subject really has the diagnosis. Once the diagnosis is confirmed is when they enter into a natural history project, and then data extraction and source document processes continue afterwards. Next slide. And when determining natural history eligibility, the genetic counselors look at two components. So one, do they have a diagnosis? So those with confirmed or presumptive diagnosis are eligible for natural history. Presumptive when there is a variant of significance identified in the genetic report, but and they have clinical features suggestive, but it has not been reported as pathogenic in the past. And then when there's only biochemical tests suggestive of the disease, or when there's an affected family member or research-based testing that is suggestive of the disease. And the next criteria that they look at is secondary diagnosis. So we don't want to have diseases that have overlapping clinical features, which will limit our ability to study the disease of interest. So they also exclude subjects with secondary diagnosis with overlapping features. Next slide. So now kind of like more in depth about the processes as it relates to ADLD will be covered by Dr. Radhika. So I'm just going to jump into applications, essentially why is a natural history study important. And I'm going to use a pediatric onset leukodystrophy called the TUP4A-LD, and how we leveraged the data we had to really get a better understanding of the disease. So TUP4A, just for context, is a hypomyelinating leukodystrophy caused by toxic gain-of-function mutation in the TUP4A gene. The subjects predominantly present with developmental delay, after which they have neuromotor regression, so loss of skills, and cognitive impairment as well. Systemic complications have also been reported in the disease. So in our natural history cohort, we have 216 participants, predominantly male and white. And the age of onset was predominantly around 9 months of age on average, with onset spanning between 0 to 12 years of age. Next slide. So once we had the complete cohort, we looked at the different variants within the cohort. What you see here is the TUP4A chain. So we plotted the variants at different regions of the TUP4A gene. And here, this is a plot where the numbers within each circle shows the number of times that the particular variant was identified. And so if you see there is one variant, which is called the D249N, which has been reported 46 times. And so that was the most common variant that was identified, one of the common variants that was identified in the cohort. And then we were able to identify all other variants as well. Next slide. So once we identified the different variants, we wanted to go in and look and see if, one thing that we want to know is when a patient presents in the clinic, we want to know what their outcomes are going to be, when they're going to be developing the different symptoms. So we went and looked at early features, so the disease onset, genotype, and then the most common presenting feature which is milestone acquisition. And we found that genotype, milestone acquisition, particularly independent sitting and onset, were predictors of disease severity in TUP4A leukodystrophy. What we see here is patients is a decision tree, essentially, which shows that patients who have that common variant we saw in the previous slide, D249N, had a probability of 75% chance of walking by 3 years. But when patients did not have that particular variant, but they were able to attain independent sitting by 9 months, their probability of walking by 3 years was 69%. And when they did not attain independent sitting by 9 months, their probability was just 13%. So there was a significant drop in their probability of sitting. Based on this, we were able to identify 3 coherent cohorts. So there was the early infantile cohort. This is the 13% that we saw here. So who are more severely affected. And then there was another cohort who gained the milestones. So that's the subjects with the D249N variant, who gained the milestones, but later had a regression in their juvenile or adult phase. And then there was a cohort, who did not have D249N, and they sat at 9 months, and their onset was over after one year of life. Next slide. So we then went in and looked at the gross motor function and communication function longitudinally over time by those 3 cohorts that we identified. Dr. Radhika will be covering more about how we identified these longitudinal encounters, but each clinic visit was evaluated to assign these gross motor function and communication functional scales retrospectively, and then they were plotted on the chart. Each row here that you see represents a single patient. And the darker shades, so reds indicate more severe manifestations, and the blues and greens represent more milder manifestation. So when we look at it here, we see that the early infantile cohort, they're very severe from the get-go, and compared to the late infantile and the D249N variants. The D249N variants, on the other hand, in terms of gross motor function, they develop all the milestones, but around the juvenile period, they start to also lose them. Compared to the late infantile, fewer patients actually end up losing milestones. When it comes to communication function, we did note that the D249N variant patients were still quite impacted in their communication function, and less with gross motor function. Next slide. We then went in, we want to then also look at what are their, systemic complications going to look like. And these are some examples. Each of these graphs show time to onset of the disease-related complication by the cohorts. The pink indicates the severe early infantile cohort, the green indicates the late infantile cohort, and the blue indicates the D249N. We saw that in terms of severity, the early infantile cohort, which is the most severe, the time to onset of these complications were always much earlier compared to the other 2 cohorts. But then, except seizure, with all of the other clinical complications, we saw that the D249N the complications were more quicker in the D249N than the late infantile cohort. So this is really helping us predict when, based on these early features, when we can expect some of those symptoms to occur. And we're also able to generate a comprehensive panel of systemic complications that we need to look out for, and that will help us, in the context of clinical trials, distinguish which is like trial intervention related versus which is really disease related. Finally, just to highlight that, since this SOPs and all the procedures that we have is designed to really mimic a clinical trial epoch, they really have regulatory standard, they follow regulatory standards as provided by the FDA. So we are able to use this cohort as a non-concurrent control arm in clinical trials. And these are examples of, when they have been used in some of our other pediatric leukodystrophies. Next slide. So just as an overview, natural history studies are able to help us understand the disease, disease presentations and progression across the lifespan. It helps enable early diagnosis, inform standard of clinical care, and predict clinical outcomes as well. In terms of clinical trial readiness, we're able to stratify cohorts for clinical trial design and also select and validate appropriate clinical outcome assessments for the clinical trials. And then we're able to identify comprehensively all disease-related complications. So we're able to discriminate them from adverse events in the, that we may see in the context of a clinical trial. And we're also able to develop a robust standard longitudinal natural history data cohort. Next slide. Finally, just key takeaways. So natural history studies require a very premeditated, standardized approach, especially when we are basing that on real world evidence. And they complement the concurrent natural history study to characterize pre-diagnosis disease course. We're able to recruit large cohorts, minimize selection bias that we would see if we only had a concurrent natural history study. And then we're able to use this data for regulatory submissions since they mimic a clinical trial epoch. And we're able to also inform future prospective studies, including clinical trials. And we're able to apply this across leukodystrophies and rare diseases, and with just minor modifications to the nuances of the rare disease being studied. Next slide. Finally, I'd like to thank our leukodystrophy patients and families, Dr. Vanderver, Dr. Adang, who are PIs of the study, Dr. Gavadze, Omar Sharbini, all the GLIA CTN sites, our external collaborators, the CHOP Leukodystrophy Center, and ADLD Center in Mayo for giving me the opportunity to present the work. I hand it over to Radhika.
Okay, thank you, Anjana. I'm going to apply all the principles and general discussion on natural history study to ADLD today. Next slide. We all understand what the disease looks like in a patient, but briefly, this disease starts in the 4th or 5th decade with signs and symptoms of autonomic dysfunction listed here, and there is subsequent motor and cerebellar impairment. The brain and spinal cord MRI findings can precede clinical manifestations by decades. We can go to the next slide. I'm going to show Dr. Cousin's previous slide here briefly, just to discuss how concurrent or real-world natural history studies are going to be key in ADLD. And the reasons are listed here because we want to maximize participation as no travel is required. We're collecting tons of data from which is longitudinal but retrospective in a short amount of time. It is enabling us to study patients from all over the world and general knowledge to help design a prospective natural history study and future clinical trials can also be understood from a chart review study. Next slide. Real-world data collection using chart review allows us to retrospectively collect data for many years, can include patients from varied locations, can also include patients that have passed away. Next point. And then we can periodically review this data, even though it is retrospective. Next slide. Again, this is a slide you have seen before, but I'm going to now repurpose it for the purpose of ADLD. As Anjana mentioned, we are leaning into GLIA to help us study ADLD. They have a vast experience with all kinds of leukodystrophies, mostly pediatric onset, but we have been able to modify this for ADLD. So essentially, we are recruiting individuals who have a molecular diagnosis of ADLD, which we are confirming by chart review. We're going to use SOPs that are already in place for a leukodystrophy studying, but are going to use it for ADLD, which involves reviewing their medical records, including imaging records. We have a research fellow, Ketty Mamela, who's really leading this effort of abstracting data and filling the red cap. And once the data is verified, this will be approved and locked by me, and I serve as the PI for this natural history study. Moving on. Inclusion criteria for this study is straightforward. A patient should be diagnosed with ADLD and should be confirmed molecularly. The patient may or may not have symptoms. This let us include family members of patients who have been diagnosed with ADLD. Next slide. These are some of the key variables that we're going to be abstracting. So diagnosis, demographics, how the patients presented. And encounters essentially is letting us go back in time, and each patient visit would be considered one encounter. And then we're going to be trying to apply some functional scales retrospectively to this data, and then hopefully be able to find time to event measures. And I'm going to show you some examples of how the REDCap data looks like for ADLD. So this is again from the REDCap. Each patient will be given a patient identification number, and then we're filling in their demographics. Do they have relatives that are affected? Their radiology data for symptom, features of presentation, and then encounter history, etc. Next slide. Next slide. You can click one more time, Margot, but essentially a little more detail into granularity of the database. We're going to ask a lot of questions through the database as to family history and more into demographics. Next slide. We do know that all patients would have neurologic dysfunction. So this lists all the neurologic features patients can have, and we will be abstracting this. For example, do they have spasticity, in which extremity it may affect? Do they have hyperreflexia, weakness, ataxia, tremor, falls? Do they use a cane or any gait aid, for example, a walker or a wheelchair? Next slide. Other neurologic or other features, example, dysarthria, sleep disturbance, any sensory symptoms, any mood disorders, changes at work in terms of productivity and cognitive decline. These are just examples of how granular and detailed the data abstraction will be based on our understanding of clinical features of this disease thus far. Next slide. And then I'm going to show you some examples of how each of these features in terms of the data that we're going to collect. For example, if a patient has ataxia, we're going to ask when the ataxia was first noted, and then additionally upload the documentation or outside records that would support the answer. So this is just built into our SOPs, and that's how we verify the data in the future. And these are just snapshots from the REDCap that GLIA has helped us modify for ADLD. Next slide. Again, example, if a patient is using a gait aid, for example, a cane, it's going to ask when did this start, what year, and then again, examples of from the clinical notes to support this. Next slide. Similarly, for example, if a patient is wheelchair dependent, it's going to be similar abstraction. Next slide. We do know that patients often have autonomic dysfunction. That's typically how this starts. So for example, we have listed all the GU symptoms that a patient can have, and again, whether they were noted or not noted. Next slide. And then if they were using any medication, for example, bladder spasmolytics when it was started, and then uploading all the documentation to support our answers. Next slide. autonomic dysfunction, everything we have listed here is also part of the database, again, based on our current understanding of what the clinical symptoms in patients have been. This has been built into database, so this just lists the autonomic dysfunction symptoms. Next slide. What are the challenges as we are going through collecting and abstracting data right now? The challenges, as noted previously, is just heterogeneity in data quality. These patients have been seen across centers through their lifetime. They may be missing data points, and then unclear documentation, but we'll work through these challenges. These are just inherent of any clinical abstraction. Next slide. We're hoping that this will allow us to have some form of uniformity in standard and care of these patients. This will help us get ready for a clinical trial and hopefully will serve as a control arm for any intervention trial. Next slide. Now we would want to also study ADLD prospectively, but just as mentioned before, there are several challenges in a rare disorder. Low prevalence, statistical power is going to be limited. There are lack of standardized clinical endpoints yet for ADLD, and patients may be getting some off-label treatments as supported care, and that might make a prospective natural history study harder to distinguish from a true natural history study. Next slide. Some of the other challenges include just a very long duration that would be needed because this is a very slowly progressive disorder. It could span into decades, which would make patient retention a very big challenge. The patients are widely distributed geographically. There would be travel limitations. It is a neurologic disorder, so mobility is certainly affected, and so recruitment may be difficult. We certainly would need multi-center international coordination. And then some ethical considerations because patients may not directly benefit from this natural history study, and then just managing expectations when there isn't a treatment yet available. Next slide. We would, some of the other challenges include just quality control if the patients are in different centers, and then funding for a rare disorder is very challenging. So we would need to partner with some philanthropic funding or private partnership like we are doing right now to sustain a long-term natural history study. Next slide. We would, however, still would like to perform natural history study prospectively for ADLD, and these are some of the scales that have been used in other neurologic disorders to study some of the similar functions, for example, gait balance, motor dysfunction, autonomic dysfunction that could be applied to ADLD, but they have not been yet standardized, so the challenge still remains. Next slide. So to briefly summarize, we certainly need an interdisciplinary collaboration for ADLD. Next point, it is an ultra-rare disorder with challenges inherently in a rare disorder. So our focus is really retrospective natural history study at the moment, and then hopefully work towards a prospective natural history study. There are a few additional points if you want to just click through them. I think that's my last slide. For the sake of time, I'll end here, but this slide lists all our collaborators across Mayo Clinic, across different centers, and special thanks to GLIA for letting us lean into their large leukodystrophy REDCap to modify it for ADLD to really give it a head start, and then ADLD Center for this partnership. Thank you all.